Happy Halloween, everyone. So we've got, I think, what will be a fairly short lecture today, and we're going to be looking at plant growth regulators. But before we do, let us review.
Last lecture, we looked at reproductive growth and development, and we looked at a number of steps. And we started with flower induction. And we said that most of our agricultural crops are self-inducive, so they will flower after the plant reaches some level of maturity, and that could be the amount of nodes it has or some other factor. Now, we can better track the developmental stage of our plants by measuring the amount of heat that they have accumulated over time rather than looking at the number of days that they have existed since germination or since we planted them. And this is because heat regulates metabolism, right? So plants will grow faster when it's warmer. And we said that the way that we can measure heat accumulation is something called growing degree days. And we looked at the temperature averaging method, which is kind of the easiest method. And in that method, you take your maximum temperature for the day, subtract the minimum temperature from it, divide by 2 to get an average, and then subtract from that a base temperature, which is the temperature below which no development is going to occur in your plant. And that base temperature will vary with different species. We went through some examples of that. We also said growing degree days is important because insects, you can track insect development as well, and that may allow you to do more targeted pest control. So those were our agricultural plants that are self-inducive, but then we said some of our agricultural plants respond to photoperiodism. And this is where they will go through different growth or developmental stages as a result of variations in day length. Now we have our day neutral plants, which don't respond to photoperiodism, but then we have our short day plants and our long day plants. Now our short day plants will, in this case, we're talking about flowering. Our short day plants will flower after the length of day is shorter than some critical day length, and our long day plants are vice versa. But really, as we saw, it's really the length of night that seems to be most important. And I went through kind of that complicated mechanism of how we think our long day plants this is working. Again, you don't need to know that, but just something for you guys to see. So we have self-inducive plants. We have plants, so self-inducive up here. We have plants that respond to photoperiodism. And then we have plants that respond to vernalization. And that's that period of cold that certain plants need. They need to be exposed to that period of cold in order for them to flower. Typical of our biennials. Okay. Then we talked about flower development, and really that what I want you guys to remember that we have that switch in our apical meristems from vegetative to reproductive, and that is irreversible. Once that switch occurs, you can't go back. However, our plants can abort those flowers or those flower buds if conditions become bad. Now we moved on to pollination, and we didn't go too deep into it because we looked at it in a past lecture, but that is the transfer of pollen from anther to stigma. Fertilization is the union of sperm and egg, and we said that some of our plants are self-fertile, so they can pollinate themselves, whereas others are self-incompatible, and they can't pollinate themselves. And with our incompatible plants, sometimes that leads to parthenocarpy, which is seedless fruit. And this is a result of a protein that is on the pollen, and the plants are able to recognize that protein as belonging to itself, and they will abort the seeds. Now, from pollination and fertilization, we get fruit set. And again, our fruit is a mature ovary and associated structures. Now, not all flowers become fruit. Some flowers are not pollinated. Some flowers get aborted. And what's happening here is your plant has limited resources. So if it put all of its resources into all of its flowers, into producing all of fruit, those fruit, you may either put too many resources in, your plant may die, or those fruits may not be ideal. They may be small and undesirable from a marketing standpoint. So if we're looking at this from a production standpoint, one of the things we want to be aware of is the optimal fruit set, right? So if we have too many fruit, they may, like I said, be small, undesirable. If, from a production standpoint, if you have too low of a fruit set, it may be not profitable. So you can manually thin fruit out, manually thin as you go along to try and get this optimal fruit set. Fruit growth and development, we said two important hormones played an important role, auxins and gibberellins. Gibberellins are sprayed on all your Thompson seedless grapes to increase the size of the fruit. Auxins play a role in, for example, in strawberry receptacle growth. And then after this stage, we have senescence, which is a deterioration of cells and tissues. And we can talk about senescence in terms of the whole organism, that whole organism, the tissues breaking down, or individual organs such as leaves and stems, where the organism may remain alive. If we're talking about herbaceous perennials, for example, we'll have senescence of the leaves and the stems in late fall and winter, yet the organism will remain alive. And senescence is just a natural process as a result of inherent physiological changes that are occurring in the plant. All right, so that's our review.
Okay, so when I say plant growth regulators, I'm primarily talking about plant hormones. Now, hormones are chemical messengers, and they are produced in small amounts at one site in a plant, and they're translocated to other sites where they alter plant growth and development. And we've already touched on some hormones really throughout the course, especially last lecture when I talked about auxins and gibberellins. We're going to revisit them. Now, this is called plant growth regulators. You know, this mask is not staying. Let's try that. There we go. This is called plant growth regulators because when we talk about plant hormones, hormones are natural substances that are produced by plants. But what we have done is we have been able to synthetically produce chemicals that are similar to these hormones in a lab or in a factory, wherever. And they act in similar ways. But they're not technically hormones. So we classify both those synthetic substances and these natural substances as plant growth regulators. Now, we are going to be looking at 5 main groups of plant hormones: auxins, gibberellins, cytokinins, or kinins, ethylene, and abscisic acid. And we're going to start with auxins. Auxins were actually the first group of hormones discovered in plants. The main, most widely known, most widespread natural auxin is indole acetic acid, or IAA. You'll see that. IBA, indole butyric acid, and naphthalene acetic acid, NAA, as well as 2,4-D, are also common synthetic auxins that you will see as well. Now, auxins originate in the meristems and enlarging tissues, particularly in our shoot tips. And then they move down through the plant through vascular tissues. All right, so auxins originate in your apical meristems, and they're going to move down the plant. One of the main uses or main functions of auxin is to maintain apical dominance. Now, I believe I talked about apical dominance in a past lecture. Essentially, it is this process by which the main apical meristem, that main apical bud that you see at the tips of your shoot, produces auxins, and those auxins travel down the plant and they inhibit the growth of lateral buds. Now, when that main bud is removed, we have the stopping that stops the flow of that auxin, and your lateral buds will then begin growing that you see here. So this is something that we could take advantage of in a nursery setting if we're trying to develop bushier plants as opposed to taller plants. If you remove that apical bud, that apical meristem, it could result, you'll lose that apical dominance and you can grow a bushier plant. So auxins are important for apical dominance. They're also important for phototropism. Remember I talked about that. That's the movement of our plants normally towards light. And it's auxin molecules that facilitate that. Right? So the sun or light is shining down on our plant. It results in a buildup of auxin on the darker side of the plant. And that auxin causes cell enlargement and cell growth on that side and cell elongation. And that's going to tip the plant towards the direction of the light. So another important use of auxins. Now, where we utilize auxins most commonly is in adventitious root initiation. So if we are propagating plants through cuttings. And the two common ones that we use are that indole butyric acid, IBA, and that naphthalene acetic acid, NAA. So those are synthetic, and like I said, they have an important commercial application. So typically, when you are going to propagate with cuttings, you will take a cutting, you will dip it in an auxin solution, and then plant it, because those auxins help stimulate those adventitious roots. And if you remember from when we talked about roots, adventitious roots are roots that are formed on a plant other than the main root system, right? Off of a radical or primary or secondary, tertiary root. So if they are formed off of a stem or sometimes even off of a leaf, those are classified as adventitious roots. Some other commercial applications that we have for auxins, they can be used for weed control. That 2,4-D is a selective weed killer of certain broadleaf weeds. They can inhibit stem sprouting. Remember, it's that auxin kind of helps maintain like dormant buds, so you can apply I'm going to struggle with this the entire time. Naphthalene acetic acid to the trunks of certain ornamental trees to prevent the development of those auxiliary stems, right? That would otherwise make your tree look bushy. And in micropropagation, where you take essentially just small amounts of plant tissue to propagate plants, auxin is an essential hormone that needs to be included in the culture medium in order for roots to initiate. Okay, so those are auxins. Now we're going to get to gibberellins. So gibberellins are probably the most numerous in terms of number of plant hormones. There are over 100 different gibberellins that have been discovered in the tissues of various plants. Now they are synthesized in our shoot apex and the root system, and gibberellins are also found in embryos and cotyledons, and we'll see why they play an important role in germination. So gibberellins move throughout the plant in both directions. Unlike auxins, which have that top-down flow, really from our apical meristems down, gibberellins can move throughout the plant. Now, in terms of commercial uses, one of the main uses that we have for gibberellins is stem growth. So gibberellins can dramatically increase stem length through cell division or elongation. Much more so than auxin, right? Auxin also has a similar role. We saw phototropism, but gibberellins do this much more dramatically. So we can take an ornamental plant, such as that poinsettia that you see there, which we will typically see as a smaller plant, and actually, with the application of gibberellins, grow it into a tree. We did, last year, we used gibberellins on, I think, pea seeds or pea plants and saw the difference in growth between the ones that we used gibberellins on and the ones that we didn't. They can also be used in flower initiation and to change or ensure the sex of your flowers. For the example, in cucumbers, if you apply gibberellins to the leaves of a cucumber, it stimulates the growth of male flowers. Right? And cucumbers are those plants that we talked about that are dioecious, right? So they have separate male and female flowers on separate plants. So if you are in a situation where you have a bunch of cucumber plants, you want to ensure that you have male flowers to pollinate the female, you can apply gibberellins to ensure that you get male flowers when you need them. Importantly, gibberellins also stimulate seedling germination and growth. Right, so remember our process of germination, our seed imbibes water. After that happens, a natural gibberellin produced by the embryo translocates to the alurone layer, that outside layer of the endosperm, and that kind of triggers it to start producing amylase, breaking down the endosperm, providing sugar that's going to power the growth of the embryo. So if you soak your seeds in gibberellic acid before germinating, it should greatly stimulate seedling emergence and growth, and this is done on a number of commercial species. Really, gibberellins and abscisic acid play this dual role of gibberellins promoting germination and abscisic acid maintaining seed dormancy. And it's both of these hormones that interact and eventually, due to particular environmental conditions, will result in a decrease in abscisic acid, an increase of gibberellins, and then your seed will germinate. We'll get to abscisic acid in a second. Other important uses for gibberellins. I talked about stem growth as being extremely important, but this is actually probably the most common use, and that is to increase the fruit size of different plants, particularly Thompson seedless grapes. And I talked about this in the last lecture, but practically all vines of grapes grown for table use are sprayed with gibberellins. So if you go to the store and you're buying grapes, they have most likely been sprayed with gibberellins to increase the size of the fruit. Another role that gibberellins can play is they can overcome the cold requirement for flowering. So those species that require vernalization, right, that period of cold before they will flower, the application of gibberellins will negate that cold requirement for certain species. As an example are azaleas, right? You can apply gibberellins to azaleas, and you won't need to have that period of cold for them to begin flowering. All right, so those are gibberellins. A lot of uses with gibberellins. Cytokinins. I should probably look up the proper pronunciation, but I'm already in this, so we're just going. I suggest you guys check out how to actually say that word. I think it's cytokinins. Anyway, there's over 100 natural and synthetic cytokinins. They primarily promote cell division. We don't have a huge agricultural application for them, but they are important for micropropagation. So I don't think we get into micropropagation in this course, but it is a process by which you take plant tissues, grow them in media in like a petri dish in a lab, and you're able to, from a small amount of plant tissue, get an entirely new plant. So during that process you have to apply or add certain things to the growth media. I already talked how you need auxins to stimulate root growth. You also need to apply cytokinins for shoot development. So that's where we primarily use that in a commercial use. All right, ethylene. As I mentioned last lecture, ethylene is a gas. It's the only hormone that is a gas in our plants. It is produced in actively growing meristems, in ripening and senescing fruits, as well as in germinating seeds. And it's also produced in certain tissues in response to bending or wounding or bruising of those tissues. Now, a synthetic ethylene, ethafon, is widely used commercially. And it's widely used primarily for fruit ripening. Right? You can apply ethylene gas to ripen bananas, honeydew melons, tomatoes. It is also used as a pre-harvest spray to promote uniform ripening of apples, cherries, figs, blueberries, coffee, and pineapple. Right? We want, from a management standpoint, from a production standpoint, it's easier if you're having all of your fruit ripen at a particular time. It's going to be easier instead of having to constantly go out and harvest. You can just harvest everything at one time. It'll cut down on those costs. So you can spray with this ethylene synthetic, and that's going to promote a uniform ripening of your species that you can then go and harvest. Just like with gibberellins, ethylene can also be used for flower initiation and changing sex expression. As gibberellins made cucumbers made male flowers, ethylene will make female flowers, female cucumber flowers, as well as female pumpkin flowers. Ethylene is also used to degreen oranges, lemons, and grapefruit. Sometimes you will have a mature orange that is still green, right? The fruit is ripe, it has the appropriate sugar content, but the rind is still green, due to just high chlorophyll levels. You can apply ethylene to those oranges and it will get rid of the green. Really, that's more for a marketing standpoint because people will see that and think it's not ripe. But by degreening them, it'll be more what people expect when they're going to go buy an orange. You can also use ethylene as a harvest aid. Certain fruit and nut crops are harvested by shaking the tree. So if you spray ethylene beforehand, it's going to improve the drop of the fruits or the nuts because there's an abscission-inducing effect of ethylene, which essentially just means it makes it more likely for those things to fall. It can also be used to stunt growth of certain plants as well. Okay, so that's ethylene. We're moving right along. We're now into abscisic acid. So abscisic acid is synthesized in both the leaves and the roots, and it has two major roles in our plants, and I already mentioned one of them. The first role is to regulate seed development. So abscisic acid prevents seeds from germinating too early. It maintains seed dormancy. The other important role in abscisic acid is it acts as a stress hormone when our plants, in response to cold or water stress. Right? So when we have a drought condition, if you guys recall, water evaporates from a plant called transpiration through those stomata, those pores under the leaves. When we have low water, when the plant senses loss of water, that abscisic acid is produced. It travels to those leaves and it causes those stomata to close to prevent further water loss. Right? So this is a stress hormone that our plants utilize for those situations. And those are our 5 main hormones, right? Easy lecture, like I said. Besides those 5 major categories, there's also some other additional hormones that are important. We have brassinolides, which are steroids that function in cell division and enlargement. And where they are important, why they are important is, or why we notice them, is that our dwarf plants, or plants that exhibit weak growth, lack brassinolides, right? So obviously they play an important role in cell division and enlargement. Some really cool hormones as well, salicylic acid. I think I mentioned this already in one of these lectures, salicylic acid. However, I'm pretty sure I said that is the active component of aspirin. I do remember when I talked about it. It's an important component of plant defense, and this is really cool. So when a plant is attacked or injured by a pathogen, it releases methyl salicylate, which is a salicylic acid. And now this compound is absorbed by the leaves of neighboring plants. And that absorption triggers those plants to produce pathogenic resistant proteins. Right? So it's a signaling hormone, and it actually winds up signaling neighboring plants as well that there is a pathogen attacking and they need to start producing defense against that pathogen. It's really cool. Along the lines, jasminates work similarly to that methyl salicylate, as a signaling attack when they are being attacked by herbivores. And there's also cysteamine, which is produced in tissue wounded by herbivores, and that gets transported to remote tissues and organs within the plant, and that triggers those tissues to start producing defense or potential defense chemicals against the herbivores. So all of those three all act as part of plant defense. And then lastly, I talked about auxins and gibberellins that promote growth. We also have compounds that retard growth. Now, these were developed in the 1950s primarily, and there's a number of different ones, and they keep plants dwarf-like. And we apply them to some ornamentals, such as mums, to keep them small, or azaleas. They're also used on turf grass. Now, that is it. Like I said, a quick lecture. Didn't want to go too in-depth on the hormones, but be aware of those 5 main groups of hormones and their effects and how we use them in a commercial application. Happy Halloween.